Welcome to the grand rounds this morning, and it's my great pleasure to introduce Dr. Brian Pogue. He's a professor of engineering science at Dartmouth. He's the director of master's and PhD degree program in the engineering science. His Optics in Medicine research group develops unique spectroscopy and imaging systems that allow visualization of medically relevant phenomena. The imaging technologies developed by this lab are used to visualize radiation therapy, guide surgery, and optimize photodynamic therapy delivery. He received his bachelor's degree from York University, majoring in physics in Canada, and he also master's degree from the same place. And he received his PhD degree from McMaster University in Canada in medical physics, and he did a postdoctoral fellowship at Mass General. Straight to the business. He's well-funded. His NIH reporter lists him as a PO1 project leader and also core director. And the core name is In Vivo Imaging, Dosimetry, and Tech Transfer. He also has two different other ones. One on surgical specimen margin assessment during breast conservation surgery, and the other on Cherenkov calibration for radiation therapy that just started this year. And I found 239 publications listed in PubMed. He's the president and co-founder of DoseOptics, and he was dean of graduate studies at Dartmouth from 2008 through 12, and he was the chair of NIH study section called Biomedical Imaging Technology, BMIT study section from 2012 through 14, and he's in editorial boards of 5 different journals, including Medical Physics. He's the fellow of the Optical Society of America, and also he's the fellow of the American Institute for Medical and Biological Engineering. Oh, welcome.
Thank you. Thank you. Well, it's a pleasure to be here. Hopefully I'll be able to show you something you haven't seen before. I think the images on this screen are hopefully something that is interesting to radiation oncology and is totally new, which is the idea of imaging radiation delivery with Cherenkov light. So you can see here a couple water tank videos and then 2 patient videos, one getting whole breast radiation therapy and one getting total skin electron therapy. And so for the first time, we're able to visualize the treatment beam as it hits the tissue. So that's what I'll talk about. I do need to disclose I am a founder and president of DoseOptics, and I will have about 4 slides on that. What I do want to talk most of the time about is radiation therapy imaging with Cherenkov light. And so what is Cherenkov? Cherenkov is a little tiny light signal that's emitted from high energy electrons or charged particles. And so it comes from cosmic rays. We see it in nuclear reactors as the blue glow in the water pool. Nuclear physics uses it for particle smashing and neutrino observatories. That's the key detected signal that are in most of these fundamental particle studies. There's some great papers from the 1970s where the Apollo astronauts claimed they saw Cherenkov from blue flashes in their retina, and then a whole series of Science and Nature papers sort of analyzing was that really Cherenkov light. There are 2 papers in the medical physics realm showing where they turned the lights off in the radiation therapy room and took pictures of the patient on the table and showed that there was Cherenkov light coming off the patient's head, and that was the origin of the blue light that they were seeing when the radiation was through their eye area. And the Cherenkov, we see it as blue, but it's actually a broadband spectrum. It just is peaked in the UV blue, but it's actually throughout the visible spectrum. One of the interesting things we realized early on is that the nuclear medicine world has been using Cherenkov or playing with Cherenkov for a while, but it's a very strong, the Cherenkov emission is a very strong function of particle energy. So as you get up above 1 MeV, you get much, much higher emission of Cherenkov light. And so there's a threshold, and above that you get Cherenkov emission. And so in this radiation therapy regime, we get, for every electron in the patient, we get a couple hundred Cherenkov photons coming out. And if we do a Monte Carlo simulation, there's perfect linearity between Cherenkov and dose. So from a theoretical standpoint, Cherenkov emission is part of the dose delivery process. So we got into the idea of, well, could we use this as a useful tool in radiation therapy dose imaging? And this was about 6 years ago now, but our initial idea was, well, let's just image it in water tanks. And so we put a water tank under a linear accelerator beam. And these are some of the first images that we got with a specialized camera that is intensified and captures single photons. And so this is a pretty poor image, but this is from a single pulse from the linear accelerator. And the linear accelerators pulse at about 360 times per second. If we integrate and median filter 15 pulses, we actually get a pretty nice image. So we built a system. Adam Glazer was a PhD student who built a system for sort of water tank gantry experimentation so we could rotate it and we could capture the beam as a dynamic beams. We wanted to look at both static and dynamic beams. And so this is our first video of an IMRT treatment plan in a water tank. So we're just cameras staring at the water tank, and you can see the linear accelerators arcing around, delivering its IMRT treatment. And we're capturing real-time video of the beam as it goes through the water tank. And so it's shown in blue here. And then we time integrate it to build up the Cherenkov map, or the dose map. And so we can create in real, and again, this is real time. This is not processing the images 24 hours later. This is real-time capture, real-time video, and then real-time integration to show the dose map in the water tank. So we published this in 2014. And of course, we wanted to compare it to the treatment plan. So here's the treatment plan. Here's the Cherenkov image. And we did gamma analysis using a 3%, 3 millimeter criteria, and showed that we could see a greater than 95% pass rate for this type of image. We did the same treatment plan for VMAT. With, again, this is from TG119. It's a mock prostate, sort of a arc shape with a zone of low dose in the center. And both pass this 3%, 3 millimeter criteria. So it's pretty neat. We were excited about this, and we've been trying to advance this, and this is one of the spin-offs of it. Perhaps even more interesting, though, is that it is real time. And so you can analyze the plan as a function of frames, and you can look to see where does the beam cross through the green zone, and where does the beam cross through the blue zone, and what's the dose deposition at each pot. So there's a potential tool here to help inverse treatment planning do a better job, or to quality audit inverse treatment planning systems. So we're exploring this through the company. We were interested in the idea of could we use this in situations where dosimetry is hard. And so we partnered with the people at Washington University who had a view-ray system. We took the camera on a road trip, so you can see our camera set up on a tripod here. And view-ray, of course, is a magnetic resonance-guided radiation therapy with 3 cobalt beams. So you can see the 3 cobalt beams here. So that tank, and because it's in a magnetic field, just the process of getting measurements out is harder and more complicated. It's not impossible, they certainly are doing it, but it's more complicated. One of the nice things about Cherenkov is we could image from across the room. In principle, we could image from many, many meters away. So we positioned the camera outside the 5 Gauss line and just imaged the water tank down the bore of the magnet. And so you'll see in the next, this was about 4 to 5 meters away. And so this is a sped up image. And so these are 3 cobalt beams going into the water tank. And so you can see we capture those. This is sped up 40 times. But this is the delivery of the cobalt beams in the water tank. and then this is the time-integrated dose map, again, for this TG119 C-shaped phantom. And we're just working with the WashU group to verify this and submit this for publication in medical physics. We, of course, did standard beams, so there's a nice 10 centimeter square beam, and we looked at the lateral profiles and the depth dose curve, and in almost all cases, the Cherenkov does a very good job. We're seeing slight discrepancy at the smallest beams, doing a bunch of Monte Carlo simulations to try to figure that out. But it's looking like this will be a useful tool for complicated situations like MR-guided radiation therapy. Just a couple quick technical developments. I won't get into the details, but these are important. So our concept here is that we would position cameras in the room and capture treatment delivery for a range of possible treatments at any time, and perhaps even passively have it on for all patients, like you would have the video monitor on so you could see the patient, but this would allow you to see the treatment for all patients. The key is, in fact, this is the most important technical piece of it, is the linear accelerator is pulsed, and so we gate the camera to those pulses from the linear accelerator. That makes it work. That improves the gain of the system by a factor of 10,000. For the physicists in the room, this is the slide that gets me most excited, which is we're using an image-intensified CMOS camera. And so the image intensifier is a photomultiplier tube that captures single photons, intensifies them, and then displays it. And our original image kind of looks like this, which kind of looks like a fuzzy glow in a snowstorm. But we do a bunch of time gating to the Cherenkov pulses. We amplify, we do salt noise removal and background removal, and we take a pretty crummy image that looks like this and clean it up to look like this. And perhaps most important is this is an image made of single photons. So we're in a room like this, we're actually doing single photon imaging of light coming off the patient, which is an incredible technical achievement, and it's been taking us many years to get to the point to be able to do that. Yep.
Can these techniques translate to a proton beam as well?
No. So we've spent a lot of time working on that. Unfortunately, Cherenkov emission is highly energy and mass dependent, and with protons, you have to get up to about 400 MV before you get Cherenkov emission. So although we've spent quite a bit of time trying to measure with protons, but unfortunately. That's right. Again, if you're just doing phantoms, you can turn the room lights off and you don't really have to gate. But with patients, obviously, we're looking for a system where we don't have to turn the room lights off. And that's where this has become really important, this time gating. So most of what I've showed you is 2D. And so we've been struggling with how could we turn this into a true 3D or 4D tool. And so this is a project that we're really right in the middle of now. Peter Bruja developed this idea of imaging with Cherenkov laterally and using the epid or the portal beam vertically along the axial direction of the beam and combining those data streams. So you get your epid image in real time, you get your Cherenkov image in real time, you superimpose those in real time and create a 4D verification system. So we're right in the middle of this now, but this is you can see this is a time-integrated treatment plan of that TG119 C shape. And so in real time, we capture the whole 3D volume and this was done at 10 frames per second, so it's 4D verification. So this is really embryonic at this point, but it's an idea that we've been playing with and we're developing. So, but where I want to get to, though, is imaging on patients. And so just to show you, you can image on patients when we position a camera up in the ceiling, and we image a solid water phantom, we indeed can see the treatment beam. So we just made a complicated treatment plan where you can actually see the individual fingers or leaflets of the MLC moving. And so indeed, what we most wanted to see is, can we see an individual leaf, and can we see an accident or an error in an individual leaf. And these were 2 treatment plans, one that had the correct delivery and one that had a corrupted delivery in a single MLC. And in this video, which I'm not sure is playing, was the difference. But anyway, so we can see sub-millimeter precision in terms of MLC positioning. And so we detected this error region right here in that mock treatment plan. So in 2014, we decided to kick off our first clinical trial, pilot trial, imaging women who were getting whole breast radiation therapy, because we realized that we could image from tissue phantoms, and in fact, we imaged from a whole bunch of phantoms with blood in them, and even the, I won't get into this, but there's interesting spectroscopic implications because the color of the light that comes out is affected by the blood oxygenation. But anyway, light comes out of tissue, and it's largely near infrared to red. So in 2014, we kicked off our first pilot trial, imaging 12 women who were getting whole breast radiotherapy. And this is the very first patient we imaged. And you can see her breathing. You can see, so a couple interesting things. You can clearly see the outline of the beam on the chest. And this was interesting to us. You can see the blood vessels in the breast because they are absorbing the Cherenkov light. And so from one aspect, that's unfortunate because it means the tissue optical properties affect the image, and it's not going to be just directly proportional to dose. But you can also think of these as sort of biological fiducial markers that actually are a way to verify that the beam is in the right spot every time. So those are some of the ideas we've been playing with. But this is, every time I present this, I am a little taken aback because I realize this is the first time ever that anybody has been able to image radiation delivery in tissue. So I'm excited. Let me put it that way. We imaged this patient. She got a standard plan where she had 6 MV treatment and a 10 MV treatment, and then 2 boost beams. And these are dynamic wedge coming in here. You can see, I'll just let this cycle through, but you can see that, you can see the treatment plan for all the different beams. And let me see if I can, okay, oops. There, this is the set of videos for the 12 patients. Putting 12 videos on a slide is always a precarious thing to do, but so you can see the 12 different beams. They're all dynamic. We captured real-time video of all the beams, and the shape of delivery matches the shape of the plan. Oops. And so we're still in the process of analyzing this data, but if we take a single 6 MV beam, for example, and match the Cherenkov to the predicted surface dose, we see a linear map between dose and Cherenkov, averaged over the whole breast. And then if we just look at it as a function of time in terms of monitor units delivered, the Cherenkov intensity is perfectly linear with the monitor units delivered. So that's been pretty exciting. The other thing we explored was this idea that because we see those blood vessels, we could do image recognition, pick out the blood vessels, and track them over time. And so for a patient that was doing a breath hold treatment, we actually see a nice flat line in terms of the movement of the blood vessels. For a patient who is free breathing, we can track the blood vessels and track the motion and be able to capture the motion in real time, again, remotely from the Cherenkov camera. And so we've gotten into this process of commercial translation. And the camera system we used costs $80,000 for a single camera. Making 2 in a room would be $160,000, and the software was research-grade software. So we spun off a company with our Dartmouth's backing, and with the goal of making cost-effective cameras, custom software for radiation therapy and packaging it for clinical trials. And that's DoseOptics, and that company has been funded by 3 SBIR grants now. And there's the main people involved in the company, largely from the engineering school at Dartmouth. Our first installation at the Dartmouth Hitchcock Medical Center was in April, and so you can see the camera up here on the wall. This was a joint partnership with Vision RT, so we're installing it, capturing the Vision RT image in real time as we capture our Cherenkov image on the patient's tissue. And so here is one of the first patients. We've done 2 so far, but you can see this is the Cherenkov image as the camera actually sees it with almost no signal processing. This is the entrance side, exit side, and then the other beam. Here's the entrance side and exit side. And so you can clearly see the beam lines on the patient's tissue, and you can clearly also see the blood vessels and other biological markers on their skin. What we've been working on is importing the dynamic log file and displaying the Cherenkov image superimposed on the surface from the CT scan, and then displaying that beside the planned dose. So here's the planned dose, or planned beam delivery with you can see the MLCs here, and here's the Cherenkov image. These 2 videos are slightly out of sync, but so you can see I think the goal we're working on is, can we do verification for these patients? One of the interesting treatments, we captured a single MLC, so the system is sensitive enough to capture a single MLC slice on a patient's skin. We're working on cranking the sensitivity of the camera up slightly to better delineate this, but indeed you can see the shape of the beam and capture things as small as a single MLC leaf. And so this is the current user interface. We're just about to enter our first clinical trial at Dartmouth in probably a month or so, where we display to the radiation therapy staff, treatment therapy people, the live Cherenkov, the cumulative Cherenkov, the treatment plan, and then the cumulative plan as it's happening. We're still going back and forth, I would say, with what's the right display software to make it maximally useful to the radiation therapy team. One of the things that we are struggling with, but we know is probably important, is capturing the patient's body, the surface map of the patient's body as it moves, and superimposing the Cherenkov on that so that we really display what is going on with the patient. Vision RT, I assume people here are familiar with Vision RT, captures the surface map of the patient, and so they get an estimate of where the body position is. We can superimpose on that the Cherenkov image, but we need to be able to use the Vision RT map to superimpose, and they only get a partial map, so we've been trying to, we've been trying to display that on the patient tissue, and I don't know if you saw this video, which I'm having struggling with to play, but the beam was wandering on the patient's tissue there, and that's because of her breathing. This is kind of a reverse, though. We've moved the beam on the CT scan. What we need to do is display the CT scan as she was really positioned with the static beam. But you can see this is not a static treatment. This is a dynamic treatment where it's modified by the patient's movement. And that's what we're trying to capture in real time for the therapy team. Last clinical trial or pilot trial I'll show you is we got into total skin electron therapy. This was really interesting for us because we saw something that we think was clinically significant for the first time. So this is where patients stand in 6 different poses, the Stanford poses, and are delivered electron therapy to their total skin surface. You can look at this patient right here, and one of the things you immediately notice is that the hands and especially the feet are colder than the rest of the body, and that's not a surprise, but they're significantly colder than the rest of the body. And so we did a bunch of TLD measurements where we taped TLDs onto the patient on many different sites, and indeed found that the dose to the wrist and to the calf was significantly lower than they were expecting. Jacqueline Andriozzi is a grad student in our group. She did a detailed analysis of this on 3 different subjects and found, in fact, that we were underdosing. The beam angles we were using were not correct, and so she won the Young Investigator Award at the American Association of Physicists in Medicine last year, 2016, for recognizing this. And to me, this is an illustration of you can think you're doing the right thing, but if you don't have the right tool to measure it, you don't know. Unless you take a picture or an image, you don't really know. And so we've published a paper in Medical Physics where we shifted the angles that we use for the beam, and most importantly, depending on which room you're in and which machine you use, you need a slightly different set of angles. And there's even some reports now suggesting that the floor material that are in the room would affect the dose at the lower end because of electron scattering off the floor. So we're looking into that. So in that paper, there's a prescription for angle spread versus SSD between the patient and the machine. Oops. I'm going to summarize a little bit now, and before I just show you one last little snippet. I recognize this is very embryonic technology in radiation therapy, and the strengths of it, though, are that it's a real-time video of the delivery. It is related to and captures patient position and delivery integrated together, and then has the potential for very large fields of view. Those are the strengths of radiation therapy imaging. The weaknesses are that it's just surface imaging. There are tissue corrections needed. We're working on this. And it's a single 2D view of the patient or the water tank. But the opportunities that we've been looking at, and I welcome other ideas where this technology could have value, is in dynamic imaging, 4D Cherenkov EPID dosimetry of water tanks, MRI radiation therapy. Those are the areas that we think make a lot of sense. And then the opportunities for beam delivery verification in patients are things like soft tissue imaging and mapping. Breast, head, and neck are the 2 pilot trials we've tried, and total skin electron therapy is the other trial. We are interested and very interested in future clinical trials, things like verify the delivery accuracy in fractionated radiation therapy. This is probably what the company will pursue, which is the idea of, can we just verify that every fraction is the same? And this is passive and doesn't need to be turned on, and so it could be done without any intervention from the radiation therapy team. That's perhaps the most important thing. We're also interested in working with Vision RT about confirming the value of patient surface mapping tools, combining surface mapping with Cherenkov for delivery verification, total electron therapy, and potentially is this the type of thing that will help incident reporting, which is real-time capture of every treatment all the time. So I'll end there and just say, this idea of Cherenkov light imaging is readily done with the room lights on. The technical key is the time gating and background noise suppression. Human dose delivery imaging captures dynamics. It has the potential to be always on. It's sensitive down to a single MLC, and we can overlay the image on the anatomy or the plan. We think there's been a lot of interest in total skin electron therapy. I realize this is not a common delivery procedure, but it's one where the dosimetry is particularly problematic. And so it's good for large area field of treatment, and we've done some discovery of the non-optimal angles. We are interested in trying to turn Cherenkov images into a calibrated dose image. This is work that's ongoing in the lab, and we have been on this pathway to translate it through DoseOptics into a system that will be hopefully available to the radiation therapy world more broadly fairly soon. And then this idea of molecular imaging with scanned radiation sheets is, again, at the laboratory level, but really I find scientifically interesting. So the people who have done most of this work are David Gladstone, head of medical physics, Leslie Jarvis, radiation oncologist, and Colleen Fox, another medical physicist. And we've had some great collaboration from Jorg at Harvard and Olga and Harold at WashU, and the people at DoseOptics, and some terrific PhD students here. So thank you.